Hello students, in our yesterday's class we discussed about some inorganic elements, inorganic molecules and inorganic compounds, how you extract from chemical analysis method as well as experiment, and one more is destructive experiment. At the same time, whatever the chemicals we get from those two experiments, when we provide to the chemistry and biology as well as physics, so how they are look, that is according to the chemistry point of view, they always concern with the functional group like aldehyde, ketone, then carboxyl group, hydroxyl group, etc. As we know that. The same time, when we give that chemical to the biological people or biologists, they always concern with two things. One is their functional aspect, and second one is whether they belongs to the protein, we can call it as amino acids. Second one is, that is whether they are belongs to the lipids. Third one carbohydrate, and fourth one is nucleotides. This is according to the biology people. Same time, when we provide those chemicals to the physics, so they always bother about, they always concern with their dimension. Clear this? This is one. And next topic was actually, that is amino acids. So what are amino acids and what mean by alpha amino acids? I told you yesterday, that is one single carbon contain both acidic group as well as basic group, simply considered as alpha amino acids. So in amino acids, or in a typical structure of amino acid, we will be having four different groups, such as one will be carboxyl group, another one is amino group, third one is hydrogen, and fourth one is aryl group or variable group, simply represented by capital R. So in the same time, I told you yesterday, name of the amino acid is based on the what type of chemical present in the R group, such as if hydrogen is present in the R group, that we can represent it as a what? Glycine. If it is methyl, that is CH3, we can represent it as what? Alanine. If it is CH2OH, that is hydroxymethyl, we can represent it as a what? Serine. The same time, when you take any amino acid, so the nature of amino acid can be changed according to the medium. According to the medium. Say for example, I mean to say, we can call it as a deuterion. Yes. And apart from that, so the amino acids can be categorized into acidic amino acids, basic amino acid, and aromatic amino acids based on the number of hydroxyl, that is methyl group. Sorry, what? Carboxyl group, and one more is, that is amine group. Yes or no? So acidic amino acid example, that is I told you glutamic acid, aspartic acid, etc. Then basic amino acid lysine, and is next to what? Neutral amino acid valine, and aromatic amino acid example tryptophan, then tyrosine, phenylalanine, many are there. So this is, and today we are moving on to the next topic, that is lipids. Lipids. And what are lipids? As we know that lipids are the generally insoluble in water. Yes or no? Lipids are generally insoluble in water. So best example, when you take a drop of water and drop into the water, or take a drop of water and put into the oil, what happens? They never ever mix each other. So for that reason, lipids are insoluble in water, and they are made up of carboxyl group, and they are made up of carboxyl group which is present at R group, that is aryl group. While writing the structure of lipid, so where we are going to write R, that is variable group or aryl group, so there will be having carboxyl group. Do remember, I mean to say the carbon must be present in the lipid. So at the R, I mean where we are going to write R, that is aryl group. So there, if I write CH3, that is methyl, we can call it as methyl. If I write C2H5, we can call it as ethyl. Yes or no? Methyl and ethyl. If instead of R, that is variable group, if I write CS3, we can call it as methyl. If it is C2H5, we can call it as ethyl. So what I am trying to tell you is that in case of lipids, I mean to say definitely lipids are made up of fatty acids, as we know that is definitely they are made up of fatty acids. I mean to say that compare the word with the protein. I told you, so the protein nothing but they are the long chain of or polymer of amino acids. Likewise, what are the lipids? Lipids are the long chain of fatty acids. Then what are fatty acids? Simple. When lipid undergo series of metabolic process, finally it leads to become fatty acids. So that fatty acids nothing but these are the end product of lipid metabolism. Fatty acids are the end product of lipid metabolism. So that see, one or the other way, the lipids are made up of fatty acids and these are made up of carbons. I mean more and more carbon atoms. See, the few examples I can give you people. One such example is palmitic acid. That is best example for the fatty acids of the lipid. So this palmitic acid is made up of 16 carbons. 16 carbons including the carboxyl group at R group. Do remember here the 16 includes this is C, that is carboxylic acid. At the same time, one such example is arachidonic acid. Yes, one more is arachidonic acid. So it is also one of the example for the lipid or fatty acid which almost contain 20 carbons including this is C which is present in the aryl group. Do remember this. Clear this? So lipids are the insoluble in water and they are made up of or they contain the main functional group is carboxyl group at aryl group, and if the CS3 is present at the R group, we can call it as a methyl, and if it is C2H5 called as ethyl. Then lipids are the long chain of fatty acids, or polymer of fatty acids. At the same time, fatty acids are the end product of lipid metabolism. Next, lipids are made up of many carbon atoms. So that one such example is palmitic acid, another one is arachidonic acid. So these two are made up of 16 carbon and 20 carbons respectively. This is one. And next simple topic is we'll be having what? The lipids. Lipids are made of, as I told you, fatty acids, and also they are made up of glycerides. They are also made up of glycerides. So the word glycerides again, so based on the number of glycerides, the lipids can be classified into monoglycerides, diglycerides, and one more is triglycerides. Do remember, monoglycerides, diglycerides, and next one is triglycerides. So these glycerides are simply considered as trihydroxypropane. So do remember, in the examination they are going to ask, or the CT or in the NEET. So that is, the lipids are made up of fatty acids and glycerides or glycerides. So the number of glycerides can be again represented as monoglycerides, diglycerides, and triglycerides. So here this glyceride is also represented as trihydroxypropane. This is one topic. At the same time, when we have lipids, so sometime, do remember when I write lipid, so this lipid is made up of only fatty acids. Do remember, sometimes the lipids are made up of fatty acids, sometimes the lipids are made up of fatty acid as well as glycerides. Do remember, sometimes it is also made up of fatty acids as well as glycerides. This is one of the thing. The same time we will be having when you take glycerides, or we can take lipids. Here we will be having two. One is fats and oils. We have some classifications. I mean to say, when we have fat or fatty acids, two classification. One is fats and oils. Yes or no? Fats and oils. So this classification, do remember, examination point of view, this fats and oils, this classification is mainly based on their melting point. Based on their melting point, do remember. So only based on their melting point we are going to classify the fats and oils in the glycerides or in the lipids. So in this, what we need to understand is the oils will be having low melting point or lower melting point. Do remember, very important. Oils have lower melting point. So for that reason, even though in case of winter season, oils remain oil. Yes or no? So when you take coconut oil, so definitely it will become, which contain fat, it will become what? Yes, solid, semi-solid or solid. But cooking oil, it always remain oil itself. Yes or no? So this is one classification. At the same time, when we have fatty acids, again we have two classification. One is saturated and one more is unsaturated fatty acids. Do remember, when we have fatty acids, so the first one is saturated fatty acids, another one is unsaturated fatty acids. So here fats and oils, this classification is based on melting point. Same time here saturated and unsaturated fatty acids, this classification is based on, that is, number of bonds present, mainly based on the number of bonds present. Here the saturated fatty acids is nothing but single bond. Clear this? Carbon to carbon, single bond, like this is considered as saturated fatty acids. Whatever, see here, we can take palmitic acid or we can take arachidonic acid, whatever the lipid you take. So where at least one single, all the carbons are attached with a single hydrogen bond is considered as saturated fatty acids. So in case of a fatty acid, if the carbon to carbon, if double bond or triple bond, is considered as unsaturated, that is fatty acids. Clear? So this is another topic. And again, when we have the lipids, sometimes the lipids or the fatty acids or glycerides contain phosphate, phosphorus, and one more is you can take phosphorus. So for that reason, this phosphorus is present in the lipid. We can call it as phospholipids. Yes, phospholipids. See, very simple. If protein is attached with a what? Lipid is called as, simple, I can tell you people, protein plus lipid, we can call it as lipoprotein. Yes or no? Lipoprotein, very simple. Protein plus lipid, this combination is considered as lipid, protein, lipoprotein. If protein plus carbohydrate, we can call it as peptidoglycan, which is present in the bacteria, peptidoglycan. So here, as I told you right now, that is, if phosphorus is present in the lipid, we can call it as phospholipids. So as we know that it is already present in the where? It is in the cell membrane. Yes, like this. Just imagine the structure, you can understand. Just imagine the structure. See, as we know this. Yes or no? Here the heads are made up of phospholipids. Yes or no? This is what. And for this one such example is, better example is lecithin. This lecithin is a lipid which contain phosphorus. So for that reason, for the phospholipids, one such example is lecithin. This is about what? Lipids. Yes. See, quickly I can revise. Lipids are the insoluble in water. They contain, that is, carboxylic group at R group, and if the CH3 is present at R group, called as methyl. If C2H5 is called as ethyl. So the lipids are made up of fatty acids, and one such example is palmitic acid and arachidonic acids. They contain 16 and 20 carbon atoms respectively, and lipids, they are made up of fatty acids as well as glycerides. Based on the number of glycerides, they can be classified into mono, di and triglycerides. So, and these glycerides can be represented as trihydroxypropane, and lipids can be classified into, that is, fats and oils. Another one is saturated and unsaturated. If lipids contain phosphorus, is considered as phospholipids. They are mainly present in the cell membrane. For that, one such example is lecithin. Yes, and till now we completed, that is, the introduction to the biomolecules. Second one is chemical analysis experiment. Third one, destructive experiment. Then amino acids as well as. So here we completed lipids. So now we are moving on to the nucleic acids. Very simple topic, that is nucleic acids. Yes, nucleic acids. Very simple. Those acidic molecules which are present in the nucleus is considered as nucleic acids. So those nucleic acids are DNA and RNA. DNA and RNA. So these DNA and RNA, when we write the structure, yes, this is what? The structure. Here we will be having SOH and OH. At first, second, third and 4 CH2OH, that is fifth. So when we write the carbon number, that is 1, 2, 3, 4, 5. Here in this structure there are 5 carbon atoms are there. There are 5 carbon atoms are there. Hence it is called as pentose. Pentose. Pentose, hexose, hepta. As we know that, in that the structure which contain 5 carbon atoms are there, so called as pentose. So called as pentose sugar. Very simple, pentose sugar. The pentose sugar is also called as ribose sugar. Clear? Pentose sugar is also called as ribose sugar. I mean to say that the sugar present in the DNA and RNA or in the nucleic acid is called as ribose sugar. Clear? Ribose sugar. And here at second carbon atom, very clear, at second carbon atom, if there is a OH, is called as RNA, called as ribonucleic acid. Is called as ribonucleic acid. Very simple. At the same time, when we write the structure of DNA, yes, same structure. Here we will be having 6, that is CH2OH. 1, 2, 3, 4 and 5. At second carbon atom, if I write only H, can understand this? See the difference. Here we will be having OH group and here we will be having H group. So when we remove the oxygen from second carbon atom, I can write deoxy. Simple, very simple, deoxy. Here the word deoxy means removal of oxygen is called as deoxy. From where? From the ribonucleic acid. So this is called a ribonucleic acid. From the ribonucleic acid we are going to remove oxygen, so called as deoxy ribonucleic acid. Yes or no? So called as DNA. Very simple to understand. So only the fundamental difference is that at second carbon atom we are going to remove O, or oxygen is considered as DNA, called as deoxyribonucleic acid. If the oxygen is present at second carbon atom, is simply considered as ribonucleic acid. This is one thing. And next thing is, when you take nucleic acids, these are the long chain of or these are the polymer of, these are long chain of or polymer of nucleotides. Clear this? See what mean by DNA and RNA? Their expand form is that is ribonucleic acid and deoxyribonucleic acid. Very simple. Here what mean by DNA or what mean by RNA? These are nothing but long chain of or polymer of nucleotides. So, say for example, protein, long chain of amino acids; lipid, long chain of fatty acids or glycerides. Likewise, DNA or RNA means long chain of nucleotides. First we need to understand what mean by nucleotides. Yes or no? What mean by nucleotides? So here, when we understand the nucleotides, means before understand the nucleotides, first we need to understand what are the ingredients, what are the different chemical composition is present in the DNA and RNA. So that is the most important concept here, the composition of DNA. Composition of DNA. First one is pentose sugar. So as we can understand, I told you many times, here is CH2OH. This is called as pentose sugar because 1, 2, 3, 4 and 5, there are 5 carbon atoms are there in the sugar, so called as pentose sugar. Is pentose, pento means 5, so there are 5 carbon atoms, hence called as pentose sugar. And second ingredient or second substance or the chemical present in the what DNA or RNA is, that is nitrogenous bases. Very important, that is nitrogenous bases. So there are 2 nitrogenous bases, one is purines, second one is pyrimidines. Purines and pyrimidines. So here again the purines are of 2. One is adenine, guanine. So here adenine is represented as A and guanine is represented as G. Very simple. Then pyrimidines again are of 2 types. One is cytosine and thymidine. Clear? Thymine is cytosine and thymine, and these are represented as C and here T. And one more thing to understand is, in case of DNA, I mean when I write DNA, the thymine is present. The same time, if I write RNA, instead of thymine, it will be having uracil represented as U. So these are the what? Nitrogenous bases. In this, adenine and guanines are double ringed structures. Do remember, adenine and guanines are double ringed structures. As we noted, very simple. That is like this. Yes, this is called as double ring structures, and cytosine, thymine and uracil are considered as single ringed structures. Clear this? This is second ingredient, and third ingredient I can write here, that is phosphorus. Phosphorus, it is in the form of phosphate. It is in the form of phosphate, that is HPO4. That is HPO4. So these are the 3 main important fundamental composition, the chemicals present in the DNA and RNA. So how they are arranged, so what kind of fashion of arrangement is there, we'll discuss in tomorrow's class. Yes, thank you very much.